Welcome back, everyone, and thank you for joining the Fielding School of Public Health's Continuing the Conversation webinar. Exposure to pesticides has increased, and the many potential effects of these pesticides are now fully known. In this webinar, Dr. Beate Ritz will talk about how she used genetic and geographic information to study links between pesticide exposure and risk for Parkinson's disease. Her research is uncovering important gene-environment interactions. That is, the ways in which genetic background and environmental exposures can influence a person's susceptibility to neurodegenerative diseases like Parkinson's, childhood cancers, and autism. If you have a question during or after the lecture, you can type it in the field at the bottom of the chat window, and that's in the bottom left-hand corner of your screen. Okay, Dr. Ritz, let's get started.
Okay, so today I will be talking about agents called pesticides that we introduce into our environment intentionally in order to kill biologic organisms. We are trying to kill either plants, those are called herbicides, or we're trying to kill pests, insects, and funguses. And so they're intentional killers that we put in the environment for biologic organisms that since we are a biologic organism as a human, might also be toxic to us. Some of them are actually neurotoxic, and those are the ones I will be addressing today because I'm going to be talking mostly about Parkinson's disease. Some of them are developmental toxins, and that's important when I study autism or I study neural tube defects in children, and those exposures mostly happen during pregnancy and very early development of the child. We also have some studies ongoing on childhood cancers. Again, the exposures there are of most interest are during pregnancy and very early in life. And some of the pesticides are known as so-called endocrine disruptors, meaning they actually affect hormonal systems in the embryo and also in the adult. They may have adverse birth outcomes, and they may have a lot of other effects on the hormone system in human beings and everything that the hormone system is linked to. Some pesticides are immunosuppressants, which then links us back to the cancers. All of the exposures to pesticides we encounter are actually involuntary, but they also occur in quite a variety of different settings. And these different settings are actually the diet. So we know that quite a few of the dietary items we consume on a daily basis contain some kind of pesticides or pesticide residues. And the NHANES biomonitoring survey, which takes a random sample of the U.S. population, finds pesticide residues in lots of the urine samples at certain points in time, again and again. It seems to be decreasing, but it's usually decreasing for one agent and increasing for another because pesticide use is changing. We also use home pesticides in the home for indoor or on our gardens and in our gardens and on our lawns. Actually, at one point, 85% of all U.S. households had stored at least one of these pesticides. And then if we are living in the Central Valley of California or in other places where agriculture is prominent, we actually also are most likely exposed to drift from these agricultural chemicals inside the homes near where crops are and where applications occur on crops. We also can take these pesticides on our shoes into our homes when we walk across roads that by accident have been sprayed, or farm workers bring them home on their clothes as well to their families. Of course, the most exposed people are farmers and professional pesticide applicators and some of the pesticide production workers. But the people we are mostly talking about today will be those who are exposed as so-called bystanders because they live and work near agricultural applications in the Central Valley. So the farming environment has, for Parkinson's disease, been discussed for 20, 30 years now, and there are quite a lot of studies showing that there is an increase in risk if you are living in rural environments, if you are farming, or if you have been drinking well water. And those are studies worldwide, but also U.S. studies that show these more non-distinct increases in risk for very broad categories. And a meta-analysis in 2000, also of studies worldwide, suggested that there's a twofold risk increase if people report that they used or were exposed to any time in their life to a pesticide. These are broad categories, and we need to ask, are all pesticides equally toxic, or should we hone in on certain pesticides, and can we explain what actually they do in order to make us sick? So now I have to go back to why Parkinson's. That is an old age onset disease that usually the patients contract when they're 68 years on average. So very few people have it when they are less than 60 years of age. But it is the second most common neurodegenerative disease after Alzheimer's disease. And we kind of know what causes it, or better, we think we do. And that's the loss of dopamine, which is a neurotransmitter in the midbrain. And this dopamine neurotransmitter is made by a number of cells called the dopaminergic neurons that are located in that midbrain area of the brain. And you see it here where I just clicked on the cursor. You see it in an image. This is the midbrain region that produces dopamine, and that's in a normal individual, while here is a Parkinson's patient on the right. And you see there are only a few red spots left, meaning these cells are actually, most of these cells that we see in the normal patients have died and do not produce dopamine anymore. We know that when about 60 to 70% of these cells have died, that's when the motor symptoms occur. But there is a period of maybe 10, 15 years prior to the motor symptoms, which are tremor, rigidity, akinesia, balance problems, lots and lots of problems with walking and standing and getting up, etc., that patients exhibit. Prior to that, we have actually what's called the non-motor symptoms. And they start, as I said, maybe a decade, maybe more earlier, and are very disabling over time as well. They include sleep problems, they include peripheral nervous system problems, heart problems, and mostly what patients complain about a lot is also depression and later in stages of Parkinson's dementia. So a pathologic hallmark of this disease is what is known as the Lewy body. And here's a Lewy body in one of these cells. And here are many, many Lewy bodies in a dopamine cell. And those are important because they help us look at the pesticide biology interaction. These Lewy bodies are made out of what's called synuclein, or alpha-synuclein, a protein that we find in every cell of our body, but in the dopaminergic neurons and in other neurons as well, they can agglomerate into these Lewy bodies, which are clumps of protein. And these proteins are probably causing some of the dysfunction of the cell that in the end causes these cells to die. And this pathology is thought to start low in the brain, or actually even in the peripheral nervous system, maybe even in the gut or in the nose. And then this pathology of Lewy bodies, of this protein, moves through the brain, arrives at the substantia nigra in the midbrain where the dopamine neurons are, and then goes into the cortex, and that's probably also when we see dementia in patients occurring. So why is all this important? It's important because what I am studying in my studies are interactions of the pesticides with systems in the cell, in the dopamine cell, or in peripheral cells that also produce some dopamine, that interact with this agglomeration, with this inclusion body formation in the dopamine neurons. And it's thought that we have a normal protein, that might, under certain stressful situations or because of genetics, misfold. These misfolded proteins are quite toxic. And they can either be going into these Lewy bodies, or they can go down a pathway to protein degradation. And we have here the proteasome, which is kind of a garbage disposal that produces these snippets of degraded protein, and they are not harmful anymore. We also know that these proteins can misfold when there's a lot of oxidative stress in a cell. And we know that some of the pesticides may actually act on oxidative stress in the cell. Some of the pesticides actually affect the mitochondrion, which is kind of the powerhouse of the cell. So they are mitochondrial toxins. And then we also know that dopamine itself is actually a highly toxic agent. So the cell does all sorts of things to keep its dopamine in check so that it doesn't interact with proteins in order to make them misfolded. And so this is the cell machinery that we are looking at in order to decipher what our pesticides do and how they do it and how they kill these dopamine neurons. So gene-environment interactions. Why do we look at interactions? Well, we know from Dr. Rossman, who came up with the sufficient component cause model, that we usually don't have just one agent causing a disease. Rather, we usually have, in a person, in order for them to become diseased, we need several component causes to be active. For example, in this, and he called them causal pies. So the first causal pie we look at here is one that's made up of age. So we know that aging is really one of the driving factors for Parkinson's disease. The older you get, the higher your risk for getting Parkinson's, because there seems to also be a normal process with aging that kills dopamine neurons. And if you think that you need to get down to 60% cell death to get Parkinson's, if we wait long enough over time, all of us would have Parkinson's. So if we get to be 120 or 150, maybe we'll all have Parkinson's. So age is a definite risk factor. But then some people never get Parkinson's disease. Why? Well, the people who get it might actually have a gene A that interacts with age in some way that they get Parkinson's earlier. The cell death just occurs earlier. Then in another person, age and gene B alone would not cause Parkinson's. They also need to be male, and they might have to be exposed to pesticides. So both this patient and this patient came down with Parkinson's. However, this person would be a control and this person would be a control because we took one slice of pie out of the whole, one slice out of this whole pie, and only when this pie is complete would we get Parkinson's. So in this case, we took the gene out. And even though this person is pesticide exposed, he will not get Parkinson's, meaning not everybody exposed to pesticides will get Parkinson's. Some people need gene B. But on the other hand, when we have gene B, and we met the pie slice that said pesticides, we also do not get disease, because in order for this pie to be complete, we need both the gene B again and the pesticide. So far, we have not been able to actually manipulate our genome, right? Nobody has been successful in doing that. So our best bet would be to actually change something, and that's what public health people like to do, that's in our environment. So the pesticides, take out this slice from this causal pie, and then the person does not get Parkinson's. If we have the complete pie like here, where we have the gene and the pesticides, occurring, then we get Parkinson's. So that's the model we're going to be dealing with for the examples that you see from now on. What did we do? I wrote a lot of grants, and one of them was finally funded in 2000, in which I proposed to find all Parkinson's patients in 3 Central Valley counties, Kern County, Tulare County, and Fresno County. And why these 3 counties? Well, they use some of the highest levels of pesticides in the nation, so in the U.S. And that is because they are highly agriculturally intensive. You can see all the green is farmland, but we of course also have urban cores like these city centers. And what we did is since January 2001, we have been contacting all clinics and neurologists in these 3 counties. And since 2008, we also have a Parkinson's disease registry active in these 3 counties that helps us find patients in all of these counties and then enroll them for our studies. So the first group of patients was enrolled over 7 years until 2007, and we are currently still enrolling. We started again in 2010 with further enrollment. The first group of patients were 473 patients that we saw, out of whom 370 were confirmed as idiopathic Parkinson's. And each and every one of these patients was seen by the movement disorder director at UCLA, who drives out to these counties to see the patients because we cannot expect them to come to us. At the same time, we also went out there and collected population controls, some by phone and mail, others by showing up at their doorstep and asking them to enroll in our study after selecting them at random from tax assessors' rolls for their residences. So we enrolled about 800 population controls from these same counties that we randomly selected, and most of the results you see today will be from the 370 confirmed cases in the first round of case ascertainment. Now we are actually up to almost 700 cases, and soon we have new results based on that larger group. So why could I promise to NIEHS to do something about pesticides and Parkinson's? Well, it's a phenomenal law that California instituted in 1972 that requires all commercial pesticide users, and farmers are included here, to report on about a monthly basis their use of pesticides. So in that pesticide use reporting system that's state mandated, they report in what county, at what geolocation, which is usually a 1 by 1 mile area, they applied what kind of chemical, how much of it in gallons, how much of it in pounds of active ingredient, how many acres they treated, how they treated it with ground or aerial spraying, what they treated, so grapes in this case, and what the date of application was. And this amazing system has provided me with all of the great information you will see. However, it wasn't good enough to just know this. So because in a 1 by 1 mile area in California, in the Central Valley, you can actually find a lot of different crops. So we just talked about the way of applying pesticides, and if you look carefully, up here is a little yellow plain, and this trail is not water vapor, it's actually pesticides. And if you look down here in this image, image from another plane, you can see, if you look very closely, that there are houses down in here, and that this white spray drifts over these houses. So clearly, when these little yellow planes spray the pesticides, even if they want to spray it within this, on this field, there's always a little bit of wind and a little bit of drift that might end up spraying also the houses right next to this application. And that is what we kind of thought would happen. Here now is a map that we created over the last 10 years. We inputted a lot of land use data into our computers. So this is not a pesticide application map. This is actually a land use map. And it's computerized by many, many undergrads that we hired over the summers. And the 1 by 1 mile grid is one of these little squares you see. And you can see that in this square are different colors. The different colors represent different fields. So for example, this field is a grape field, and these are almonds, and these are kiwi, and these are field crops. So if my POR report tells me that parathion was applied on almonds in this square, then I know that the application was either here, 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 or here. And if a patient lived here, then I know he was probably close to this application on almonds. And that is very important because now when I say, well, let's go for parathion, and it was applied on almonds, you can now see that these are all the parts of this larger square that actually received application. And I would know that this house and this house had a possibility of being exposed, while maybe a house over here or here would not have been exposed. So we can computerize all of this, and I have a collaborator at USC who is a geographer, Dr. Coburn, who has made a computerized tool out of all of this. And we can now, when we ask our patients and controls what their address was, geocode the address, and then draw buffers of 500 meters or 1,000 meters around these homes, and we know exactly where the pesticide application happened and whether it might have drifted over this home or not from this field. Okay? And that's the system we use since 1974, when the first of these applications was reported to the system, up to the time when the diagnosis was made or our controls were interviewed. And we're using that to assess all the different kinds of pesticides reported to the California system. And that, of course, brings up the next question. What are these pesticides? And are all of these pesticides relevant? Are they all equally neurotoxic? And I showed you my little pie graph where I said, well, we need genes, and we probably need a pesticide. We need aging and maybe maleness in order for patients to come down with Parkinson's. So right now I want to say which of these 640 different pesticides used in the state of California are actually harmful. And where do I go? Well, I actually go to my colleagues who do animal testing. And the most extensive animal testing has been done on a product called paraquat. And that's because paraquat looks it's down here. It looks chemically very similar to an agent called MPP+, that is a metabolite of MPTP, which was a contaminant in a synthetic heroin that gave people in the 1980s instantaneous Parkinson's disease. And that was young onset people. They were 27 years, 29 years old when they injected the heroin, and weeks or months later became acutely Parkinsonian. And when later their brains were imaged, you could see that these areas where the dopamine neurons should light up did not light up at all. So paraquat looks similar to NPP+, which is the toxin that can cause Parkinson's. Therefore, paraquat was of interest to animal modelers who do testing. And interestingly, paraquat alone gives some mice and some rats Parkinsonian-like features, but a lot of mice don't get anything. And other researchers had the idea of combining this paraquat with some other agents, for example, the fungicide Monet, and found that the two combined actually would produce much stronger effects in animals. So we have paraquat that we know from animal testing can actually cause oxidative stress in some cells and some kind of Parkinsonian features in animals, and Monet makes it worse. Okay, so those are two very good candidates for testing of my system, right? And that's exactly what we did. So we used my pesticide exposure assessment system and compared case histories of where they lived, and my system then told me what kind of pesticide was applied within the vicinity of their homes and actually their workplaces, and I asked just a simple question in this first paper from 2009, what happens if they were not exposed, only paraquat was sprayed in the vicinity of the home, or both paraquat and Monet were sprayed. And you can see here that in case Monet alone was too rare, but when both Monet and paraquat exposures occurred, and you can see here that it's not rare, a lot of people were, cases, 88 and almost 50 controls, were exposed to both of these agents, then your chance of getting Parkinson's was 75% increased. That is the first data, human data ever, after the animal studies that showed that the combination of these two pesticides actually increased your risk of Parkinson's from environmental exposure. So then I told you there are lots of mechanisms in the cell that contribute to the demise of the dopamine neurons. And one of the mechanisms is the so-called proteasome dysfunction. So the proteasome, again, that's this organelle, acts like a garbage disposal. You have misfolded or damaged proteins that then are guided to this garbage disposal. And the garbage disposal does exactly what the garbage disposal is supposed to do. It chops it up into pieces. If this is clogged up, just like a garbage disposal, you have misfolded proteins in the cell more and more often, and they may find each other, they may aggregate and then form Lewy bodies, but on the way to forming Lewy bodies, they are known to be quite toxic to a cell. So one way of spoiling this system would be to do something to clog up this proteasome, right? Because then you get a lot more misfolded protein that then becomes Lewy bodies, but in the meantime is toxic. My colleague, Dr. Bronstein, developed a system to actually look at the proteasome function in human neuroblastoma cell lines that he made glow green with a fluorescent protein when they are active, and then he clogged up the proteasome with a known proteasome inhibiting chemical, and they don't shine green anymore. So when he used this system for pesticide screening, he found that there were a number of pesticides, for example, Furbam, Xyram, and our favorite Manef that we have already heard about Manef did not inhibit the proteasome, but Furbam and Xyram did inhibit the proteasome. And they inhibited it at quite low, 10 micromolar and less levels. He also found some others like Benomyl and carbendazim doing a similar kind of inhibition of the proteasome. So I used this data to see what Xyram and Furbam are doing in human populations. And Furbam wasn't used enough in my population, so I ended up just looking at Xyram. When I look at Xyram exposure at residences only, I did not see much of an effect on Parkinson's disease, but exposure around workplaces increased the risk about 50%, and if you were exposed at both the workplace and the residence, your risk increased threefold again. So we have the cell study, and we're going right from the cell study to the testing in humans, where we have our exposure assessment tools that we developed. Another thing I told you was that dopamine is rather toxic, right? And that the cell does a lot of things to keep dopamine away from proteins. Well, dopamine is being metabolized also by cells to make it less toxic. The problem is, however, that in the process of making dopamine less toxic, they create an intermediate called aldehyde, dopamine aldehyde, this one, that is actually much more toxic than dopamine itself. And the cell has an enzyme called aldehyde dehydrogenase that removes this aldehyde quickly by making this dopac compound that is not toxic. So again, we were thinking about what could increase toxicity of dopamine. Well, it's aldehyde dehydrogenase inhibition, meaning that we then have more dopal, which is toxic, in the cell that then can harm the cell more. And again, my colleague Dr. Bronstein in his lab tested aldehyde dehydrogenase inhibition for our pesticides. And here our favorite Manef actually shows up. It's this one. You can see it in this screen. So it is at even low concentrations toxic to aldehyde dehydrogenase, meaning it inhibits this enzyme, and therefore we produce a lot more of the toxic dopal agent. And interestingly, Xyram does the same. So Xyram inhibits the proteasome and also inhibits aldehyde dehydrogenase. That's two hits to a vulnerable cell. And we wrote that up in a paper recently that came out in the National Academy of Sciences proceedings. And we tested then all of the pesticides that we found some kind of aldehyde dehydrogenase inhibition in the lab for: phenamyl, endosulfan, Xyram, dialdrin, triflunomazole. And guess what? All of them were increasing risk of Parkinson's for exposures at residences as well as at workplaces. And that's part of that paper I just discussed. And there's one other pesticide I was really interested in because we know they are highly neurotoxic, and that's organophosphate. And those are actually the agents I described at the beginning of my talk as being found in the urine samples of most of the population of the U.S. And that is due to the fact that chlorpyrifos is a OP pesticide that's not used in agriculture as much as indoors until it was banned by EPA not long ago. And diazinon is another one that we also find in urine of the U.S. samples in 30% of all people because that's the one mostly used in agriculture. So those are all the pesticides of interest. So I just described the ones I was most interested in out of the 640 and why I was interested in them. So we still have to select our genes, right? And we have more than 20,000 genes to select from. And how do we do this? Well, one of the ways to look at this is, again, to go back to what we know about the biology of Parkinson's. And I told you dopamine is toxic. Well, the transporters of dopamine contribute to how much dopamine there is in the cell at what time. The transporters of dopamine also transport agents that are neurotoxic, such as MPPP that I showed you before, into cells. Therefore, dopamine transporter genetic variants are of interest, variants that cause this transporter to either be transporting more or less of the substance into the cell. Another protein and gene that codes for this protein I was really interested in was paraoxonase 1, because this is the protein that metabolizes all of the organophosphorus pesticides and therefore determines whether or not they are toxic to a person, because we have a wide variety of different reactions in the general population to OP pesticides, and that's very well known. And then finally, I'm interested in genes that determine how these proteins that deal with oxidative stress act. And I already mentioned aldehyde dehydrogenase that metabolizes our dopamine, and then also nitric oxide synthase that produces nitric oxide, which is very pro-oxidant in dopamine neurons. So right now, I will just hone into these kind of proteins and the genes that code for them and the variants that influence function of these proteins for my gene-environment interactions. So the first gene-environment interaction we looked at, we went back to our earliest interest, which was a manip paraquat exposure, and I just told you that the dopamine transporter is of importance because the dopamine transporter determines how much toxin gets into a cell or how much dopamine gets back into the cell after it's been excreted at the synapse and what it does in the cell. So there was a study at UW in Washington that suggested that there is an interaction between genetic variants of the dopamine transporter and Parkinson's disease. And so there are certain variants, and they're called PLADs and DNTRs, that put you at risk for Parkinson's. And we looked at this, and indeed it looked like we saw a little bit of an increase, 60% and 80% increase with these kind of risk variants in our study. So this was the Washington study, this was our study. We actually recapitulated what they saw for these variants. However, what I'm more interested in is actually whether pesticides now interact with this genetic this predisposition to, possible predisposition to Parkinson's. And the Washington paper had already given us a hint. So here is the scoring for risk variance in the dopamine transporter, and you can see if you're not pesticide exposed, you almost have no risk increase. And the confidence intervals are quite wide and include the 1, which is our null. However, if you're pesticide exposed in this study and you have one of the risk alleles, you have a 63% risk increase, and if you have 2 or more, your risk goes up 6-fold. When I saw this early result, I thought that's a one-in-a-lifetime finding. Nobody will reproduce that. Well, I tried to reproduce it, and lo and behold, we did. So here's the same risk allele counting, 0, 1, 2, and more. You can see that if there's no ambient exposure to manib and paraquat, there's very little to no risk. But when we have exposures at high levels and 1 risk allele, then we have a 3-fold risk increase. And if we go to 2 and more risk alleles, we actually do see the 4.5, almost 5-fold risk increase that has been shown before. So this is a gene-environment interaction that we actually replicated, and you can see that here also graphically displayed as well. The next one we looked at to see whether my hunch on OP pesticides, organophosphates, being neurotoxic is correct, was an interaction between the PON1 enzyme and OP pesticides. And what is the relevance of this? Well, we have a pesticide, and here's chlorpyrifos that I told you we all were exposed to at one point because it's been used indoors widely in the U.S. PON1 is the enzyme that on the pathway to a non-toxic or less toxic metabolite that we excrete in our urine that is being found in our urine. So we have an enzyme here, the P450 system enzyme, that actually makes an oxone, which is a highly toxic agent that is neurotoxic. And then we have paraoxonase 1 that detoxifies these oxons. So if we have paraoxonase that's not very active, then we have a lot more of these oxons that are highly toxic than somebody who has a very fast enzyme paraoxonase. And we actually know from human studies, and we've known for more than 15 years, that there's a 10 to 40-fold inherited difference in the activity of this enzyme in the human population. And that this kind of difference is mostly attributable to 2 common genetic variants in the PON1 gene. And it has been tested. One is called the 55LM, and the other is called the 192QR. And you can see that the ones with 192QR and 55MM have low serum activity for the enzyme, while the ones on the other end with the 55LL and the 192RR have the highest activity of this enzyme. So if you have low activity, you have a lot of the toxic oxones in your body when you're exposed. If you have high activity, it's actually metabolized fast. We used this to look at our patients, and this is what we saw. We saw for diazinon OP, there's almost no effect here if you're not exposed if you're not exposed to diazinon for PON1, which is what we expect. PON1 only interacts with the OP pesticide. It's not a genetic variant that causes Parkinson's disease. However, if you have the PON1 LL55 variant and the Q variant of the 192, and then you're exposed, you can see that only the people who have the risk variants of these 2 genetic variants are highly at risk for Parkinson's, about 2.5-fold risk increase, while the ones who have a very fast enzyme are almost similar to the ones who are not exposed. So that makes the difference in a population between this person who has a much higher risk of getting Parkinson's, this person who has an intermediate risk, and this person who has a very low risk, but all of them are exposed to diazinon. And when you now move to chlorpyrifos, you see exactly the same thing. Only your risk is almost 3.5-fold if you have the low activity variant of these polymorphisms. And this similarly happens also when you're exposed to parathion, another OP pesticide. So this clearly tells a very unique or a very convincing story in my mind that we need the genetic makeup to be susceptible to organophosphates. And when we're exposed, we do or do not get Parkinson's depending on how much of the toxin our body is producing and how fast our body can actually get rid of these toxins. We used the same model to actually look at household pesticide usage. We had asked all of our patients and controls to report throughout their lifetime what they had used. And we asked them for the name of the product because most people do not remember what's in their product. But then since California actually has a list of the pesticides that we are allowed to sell over the counter in garden shops, etc., we could go back to that list, back to the 1960s and 70s, and see for every product that was reported by our study participants what was in the product. So we were able to see whether they had used any household product, OP products, organophosphate, dithiophosphates, and whether they had used chlorpyrifos and diazinon. And you can see here that their risk increased quite a bit with just the plain use of reporting these pesticides in the home as use. However, when we now look at our para-oxidase interactions, and we look at people who never or rarely used any pesticides, and the people who used any pesticides, and who had either the well-functioning gene or the not so well-functioning gene, you can see that if you have the well-functioning and you're not exposed, you have no risk, or the not well-functioning and you're not exposed, you have no risk. So your risk is the same, it doesn't matter. But if you have the not so well-functioning gene, or better, protein that detoxifies, that's when your risk is almost twofold, okay? And you can see over here the same thing for another pesticide. So we see the same gene-environment interaction that we saw before for our residential workplace exposure from agricultural. We also see it for in-house use of these OP pesticides that people had reported. And the final example was for nitric oxide synthase, and since it's getting late, I will hurry up with this. Nitric oxide synthase increases oxidative stress, and we looked at interactions again with the organophosphate pesticides, and we actually looked at our household pesticide use. And again, for those people who had a variant of that gene and exposure, it was much worse than for the people who had another variant of that gene and either had used any pesticide or OP pesticides. And the same was actually true when we used our residential and workplace exposures from the agricultural assessment. So again, this is another gene-environment interaction that we were able to find because we have this very detailed pesticide exposure assessment tool that we can use. So in conclusion, gene-environment interaction studies in humans that are based on laboratory and experimental studies can be extremely useful. When we use what we know from biology, find the right genes, find the pathways that these pesticides seem to be acting on, and then put both together, we are actually able to get a much more complete picture of what's going on than when we're just randomly looking at pesticides or randomly looking at genes to try to explain this disease. So definitely we can conclude that pesticides put individuals at risk more when they have certain common gene variants that make them susceptible to either the toxicity of the pesticide or make them susceptible to have a breakdown in one of the pathways that lead to this Parkinsonian cell demise. And so the interactions of pesticides and genetic mutations are very, very relevant for biology. They make a credible story for our pesticides, much more credible than just saying I pulled one of 640 pesticides out of the hat, and it seems to be increasing the risk of getting Parkinson's disease. And a stronger scientific story is very, very important to justify and stimulate environmental regulation, and also to think about the development of neuroprotective agents that may actually decrease the vulnerability in certain people. But of course, first of all, we have to eliminate exposures that could harm them. But some people might also be helped by neuroprotection if we ever can get to that. That's my last slide, and this is an important slide. I want to thank everybody who has worked on this, and that's now 15 years of students at UCLA who wrote their doctoral dissertations and who were supported by the money coming from NIEHS for all of this work. And you can see many of my students who were involved in all of this from these pictures. But I also want to thank Dr. Bronstein, who sees most of these patients, and Dr. Bordelon, who is also seeing these patients. They drive out 4 hours to Fresno to phenotype these patients. And I think part of why we have been so successful is because we have such great phenotyping of our Parkinson's patients, and we know exactly how they do, and we even follow them over many years to see how they progress in their disease. And that's all I wanted to say.
Thank you, Dr. Ritz. We're going to do a couple of question and answers. Dr. Ritz, we have a question from one of the listeners. She wants to know, is the soil analysis something that could additionally be used to determine potential exposures?
That's a very interesting question. I'm always asked whether this exposure model, because really it's a model, right? We are not really measuring pesticides. We're assuming somebody who lived next to an application was actually exposed, or somebody worked next to an application. And soil testing of pesticides can, of course, be done. But remember that Parkinson's disease is a disease you get when you're 60 or older most of the time. Some of our patients are 39 and 40 and 50, of course. But really, the exposures must have happened 20, 30 years prior to you getting Parkinson's disease, and they probably are chronic. So the only soil testing that would be useful or helpful would be for pesticides that remain in the soil. And we know that residues of DDT, DDE do remain in the soil for 30 years. So those we might be able to find. But OB pesticides you will not find after a few weeks, after a few months. And so what does current exposure of current soil levels tell us about somebody who developed Parkinson's, probably due to exposure over a 30-year period? It's not clear to me.
Another question, Dr. Ritz. Did I understand correctly that the genes that you selected for the study do not cause Parkinson's on their own?
Yes, yes, that's actually true. The genes that I showed you here, none of them is known to cause Parkinson's disease. So the genes really are either involved in detoxifying a toxic agent such as these OP pesticides, or they are involved in proteins that in the cell, in the dopamine cell, handle toxics. And therefore, when I look at these gene-environment interactions, what I'm saying is that a single gene or its mutation or its variant actually will not put you at higher risk of Parkinson's. It has to actually come together in these kind of pie charts that I showed you with a stressor. And this stressor can be a pesticide. This stressor can also be some other toxic agent that the patient may have come in contact with throughout his lifetime.
Dr. Ritz, we have another question from a listener. To what extent do you think the predictive screens that you discussed could be used to augment regulatory review of new pesticides by the government agencies and even safer design of new pesticides by businesses?
That's a very good question, and that's of course why our colleagues who do molecular toxicology studies do this. Yeah, some of these screens should definitely be looked at because they are so relevant for neurodegenerative disorders, and EPA should probably look at them in terms of can they be used to regulate pesticides or to find out what the possible chronic effects of pesticides are. However, they, even though they show that aldehyde dehydrogenase inhibition or proteasome inhibition happens with these pesticides, we need to know we need to know these pathways, and we need to know that that's really the relevant pathway. So for Parkinson's, we know that, and that's definitely a route to go down. But for many other diseases, we actually don't know it. So we need a lot more different kinds of testings in the test tube and in animals than just what I showed you. But these are good steps to make, I think, in the future.
Dr. Ritz, can you apply this model to other diseases?
Yes, I think we can. Of course, with Parkinson's, we have a lot of knowledge, basic science knowledge that we were able to combine. And I have been working with basic scientists for more than 15 years. I think that epidemiologists actually in general should be working more with colleagues either in the clinical or the basic sciences to try to study these kind of mechanisms. And I know in cancer there's a lot of work done in a similar way to what I've been showing you. So cancer studies, definitely. There's also some efforts in neurodevelopmental studies, but for many, many diseases we're quite behind in linking our forces to actually do this.
You mentioned earlier, and I just want to see if this is correct, you say that many people are exposed to multiple pesticides?
Yeah. Yes. So that's actually a very good point. Multiple pesticide exposures occur, of course, because we are exposed through food, we are exposed through indoor use of pesticides, we are exposed possibly through agricultural use pesticides, and if we are working in these fields because we use them. So most people have more than one exposure, and it is actually really, really hard to find anyone who's pesticide exposed who's only exposed to one pesticide at a time. And what we really don't know is how these pesticides interact on maybe the same pathways or different pathways that lead to these kind of effects that then in the end lead to the disease. And we are usually in toxicology screens studying one pathway, studying one pesticide at a time, and rarely ever study combinations. So it is it's very possible that we actually need to look at combinations of pesticides and combinations of effects, combinations of mechanisms to really determine the toxicity of these pesticides, just like we have seen that paraquat toxicity is greatly enhanced when you are at the same time also exposed to manna. So we cannot say, well, one pesticide has a low toxicity, the other a high toxicity, so let's just get rid of the high toxicity one, when really truly what it is a combination of many low toxic substances that when they come together they're not lowly, they have more than a low toxicity.
Okay. Well, I don't see, and sorry.
Those are real challenges. Yeah, those are real challenges because EPA regulations also are usually based on one pesticide tested at one time.
Well, I don't see any other questions right now, so I'm going to wrap it up. I want to thank everyone for joining us today for the webinar. Next time, you can join us on Wednesday, January 15th, when we continue the conversation with Dr. Dylan Roby, and he'll be discussing the implementation of the Affordable Care Act in a talk titled Got Insurance? Where does California stand in making progress towards universal coverage? And if you missed any of our previous webinars, you can go to the Fielding School of Public Health's website, and that's ph.ucla.edu, and they're all archived there.